So in this video, we're going to talk about the intrinsic control of GFR. And so with that, we've got these two outcomes. So first of all, I need to distinguish between intrinsic and extrinsic control. So intrinsic control, there are two ways that we can regulate this. So we've got myogenic regulation, or the myogenic mechanism. Okay, that gets into autoregulation, which we talked about previously. And then something called tubuloglomerular feedback. It's a word that'll get you a lot of points on Words with Friends or Scrabble. Then we've got extrinsic control, which we'll talk about on the following video. So you've got the neural control, which is primarily the barrel reflex, which we've talked about. And then you've got hormonal control, which involves the renin, angiotensin, aldosterone system and the natriuretic peptides. Okay. When it comes to intrinsic control, the goal of this intrinsic control is to maintain GFR. Okay. Basically, changes are happening to GFR, and the kidney is doing things to make sure that GFR is maintained. Okay. Myogenic regulation responds to changes in mean arterial pressure, and tubuloglomerular feedback responds to changes that come from the macula densa in response to changes in sodium concentration. So let's talk about each of those. So remember, autoregulation is maintaining flow despite changes in pressure. All right. And so if we do something such as exercise, or for whatever reason our blood pressure goes up, this is going to cause your smooth muscle to be stretched out. The afferent arteriole is going to stretch out because there's going to be higher pressure. Well, what happens is that we want that afferent arteriole to vasoconstrict, because if it just balloons out and lets more blood in, that will increase glomerular hydrostatic pressure, which will lead to an increase in GFR. We don't want high GFR. We want normal GFR. And so by vasoconstricting that afferent arteriole, we're reducing glomerular hydrostatic pressure, and GFR as well as urine volume will go back to normal. The reverse is also true. If we've got, or someone has low blood pressure, there's not going to be that stretch on the smooth muscle. And so what happens is the afferent arteriole will just naturally dilate even more. Okay. This will allow greater blood flow through the afferent arteriole, which will increase hydrostatic pressure, which will increase GFR as well as urine volume back to normal. So we've got a picture that shows this here. So on the left, we've got kind of the cause. So notice we've got high blood pressure. It's causing that afferent arteriole to stretch. Okay, that's going to increase hydrostatic pressure, which will increase GFR, which will also lead to increased urine volume. Okay, we don't want that. We want normal. Okay, we don't want high. We don't want low. We want normal. So what happens is, notice the afferent arteriole constricts. That reduces hydrostatic pressure, and that reduces GFR back to normal. The reverse is also true if we've got low blood pressure. Okay, there's no stretch on the afferent arteriole. There's low pressure, low hydrostatic pressure, which means there's low GFR. And so as a result, what happens is we vasodilate that afferent arteriole, which will let more blood flow in, which will increase GFR back to normal. Okay, it's all about maintaining that normal flow. Okay, and this can happen if pressure is too low or if pressure is too high for the most part. Then we've got tubuloglomerular feedback, and tubuloglomerular feedback is really regulated by something called the Juxtaglomerular apparatus. Two words that will also get you a lot of points in Words with Friends, or the JGA. Okay, and so this is where the distal portion of the nephron passes between the afferent and efferent arterioles. Okay, so it kind of goes up through the middle, and you can see that here in this picture. Okay, so you've got the arterioles, and then you've got the nephron passing through. Okay, the JGA consists of macula densa, which are sensors, and then the juxtaglomerular cells. The macula densa cells sense sodium concentration. Okay, they can ultimately release adenosine, okay, which we haven't talked about before and won't talk about the rest of the semester except for in this specific context. And they also can send signals to the juxtaglomerular cells. Okay, the juxtaglomerular cells are involved with long-term blood pressure control. It's one of the things I've mentioned and will continue mentioning is that your kidneys are ridiculously important when it comes to long-term blood pressure control. The juxtaglomerular cells will release renin, okay, and then renin can ultimately causes the production and release of several different hormones that we'll get into in the next video. All right, so if we have high sodium, that causes the macula densa to release adenosine. Adenosine is a vasoconstrictor, and so it will cause the afferent arteriole to constrict. Okay? If we have low sodium, the macula densa stops releasing or doesn't release as much adenosine, which allows the afferent arteriole to vasodilate. In both conditions, this is what we call paracrine signaling. In other words, a signal is being released from one cell and it's targeting a nearby cell. That's what paracrine signaling is all about. Okay, so how does this all play into tubuloglomerular feedback? Well, if you have high GFR, you have greater filtrate flow. Okay, so just think about it, the river's flowing quickly. Okay, it's not a slow, lazy river, it's a fast-flowing river. Okay, as a result, sodium, which we talked about previously, is pumped out of the ascending limb. That sodium's moving by so quickly that it doesn't always attach to the pumps. And so as a result, there's going to be high sodium concentration at the top of the ascending limb. This is sensed by the macula densa, and so the macula densa releases adenosine. Adenosine causes the afferent arteriole to constrict, which reduces GFR, and we get GFR back to normal, we get sodium concentrations back to normal. It's an example of a negative feedback loop. Okay, so here, diagram to see. So high GFR, we increase that filtrate flow rate. It's going so fast that we can't pump out as much sodium as normal. The macula densa senses this. It releases adenosine. The adenosine causes vasoconstriction of the afferent arteriole, and that's a negative feedback loop because we're reducing blood flow into the glomerulus, which means lower glomerular hydrostatic pressure, which reduces GFR. The reverse is also true. If GFR is really low, that means the filtrate is moving slower. And so as a result, it's a lot easier to pump sodium out of the ascending limb. The macula densa senses the low sodium. And so as a result, it stops releasing adenosine, and that allows the afferent arteriole to vasodilate to get GFR back to normal. So I want to end with a thought question. So there are certain drugs that treat high blood pressure which cause vasodilation of systemic arteries and arterioles, including those of the kidneys, specifically the afferent arteriole. So if I give someone a vasodilator, what's that going to do on GFR? The answer is it's going to cause dilation of the afferent arteriole, which will increase GFR. Now, do we want high GFR? The answer is no. So how would the autoregulatory mechanisms, think intrinsic control, respond to these changes or this change in GFR? Well, the myogenic mechanism will vasoconstrict the afferent arteriole just naturally, and tubuloglomerular feedback will ultimately cause the release of vasoconstrictors, which will also lead to vasoconstriction of the afferent arteriole. And so we can reduce someone's blood pressure while also maintaining GFR. So at the end of this video, can you explain what is meant by intrinsic control of GFR? And can you explain how myogenic mechanism works as well as tubuloglomerular feedback?